Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll be discussing another topic, chromosomal aberration assay, FISH, fluorescent in situ hybridization. Let's directly go into the question. The question is, what assay is most commonly used to assess the ability of a test material to induce chromosomal aberrations in vivo? So we need to identify the assay which is used to ability to test material induced chromosome aberration. We have option A thymidine kinase assay. We have option B rodent micronucleus assay. We have option C bacterial Ames assay, and option D, that is sister chromatase assay. So the right answer will be option B, that is in green text, rodent micronucleus assay. So let's see what the American Board of Toxicology has given the explanation and reference for this particular option. The micronucleus test is widely recognized as one of the most useful and reliable tests for carcinogens that act by causing DNA damage. So we already know that carcinogens, two mechanisms will be there. One is genotoxic mechanism, non-genotoxic mechanism. So the one which are causing through DNA damage and eventually causing cancer, no, those type of can be identified by using this rodent micronucleus test. So this another thing what you have to remember is this micronucleus assay is, it is an important part of the OECD guidelines for testing chemical safety. So it is a part of this battery of genotoxic test. So now we will see what an additional reference which American Board of Toxicology has given. It has asked us to go back and look into Cassette and All ninth edition, page number 525 as well as 526. So unlike my other chromosomal aberration videos, in this, no, from the Clayson textbook, I have completely included in next subsequent four five slides will be there. So in the Clayson textbook, whatever they have given about chromosomal aberration, I have included here. So you can directly read from Clayson, or if interested, you can also go through this presentation, which clearly contains all the matter which is given under chromosomal aberration heading. So let's read and understand. Chromosomal aberration cytotoxic assay relies on the use of microscopy for the direct observation of the effects of interest. This approach differs sharply from the indirectness or traditional genetic assays in which one observes a phenotype and reaches conclusions about the genes. Okay, here unlike the previous traditional methods, here we are directly looking into direct observation and the effect of interest. So this chromosomal aberration, no, is only through addition of DNA sequencing that genetic assays can approach the directness of this cytogenic assay. In conventional cytogenetics, metaphase analysis is used to detect chromosomal anomalies, especially unstable chromosome and chromatid aberrations. A key factor is the design of cytogenetic assay. Uh, Obtained appropriate cell population for treatment and analysis. Cells with a stable, well-defined karyotype, short generation type, low chromosome number, large chromosomes are the ideal for this cytogenic assay. Okay, for this reason, Chinese hamster cells have been used widely in the case of cytogenetic assay. Okay. Um, Other cells are also suitable, like you can also use human cells, you can use peripheral lymphocytes, have been used extensively, but the preferable choice will be Chinese hamster cells. Cells should be treated during a sensitive period of the cell cycle. What is that sensitive period? Typically, it is the S phase. In the case of, we have S phase, G phase, like this. In the cell cycle, four phases will be there, no, particularly during typically S phase. Aberration should be analyzed at the first mitotic division after treatment so that sensitivity of the assay is not reduced by unstable aberrations being lost during cell division. Examples of this chromosome aberration are there in figure 9.6. So in Clayson, two pictures will be there in this presentation I will give. This is the 9.6. These are the chromosome aberration induced by X-rays in Chinese hamster ovary cells. See, if you look into picture A, it is a chromatid deletion. Okay, here you can see. And if you look into B, picture B, chromatid exchange called a triadal. Okay, here. And option C, picture C is a small interstitial deletion. Here you can see the red arrow, and that resulted from the chromosome breakage, right? And fourth picture D is a metaphase with more than one aberration. Here you can here you can clearly see a metaphase with more than one aberration, a centric ring plus an acentric fragment, here this one, right? And a dicentric chromosome plus an acentric fragment here. Okay, so these are the different types of chromosome aberrations. So cytogenic assay requires careful attention to growth conditions, control, dose levels, treatment conditions, time interval between the treatment, and sampling of the cells for analysis. Data collection is a critical period in the case of cytogenetic analysis. It is essential that sufficient cells be analyzed because a negative result in a small sample is inconclusive. If the sample size is too small, and in that if you are getting a negative result, that is not a it is the result interpretation will be inconclusive. Results should be recorded for a specific classes of aberrations, since we have seen all different types of aberrations are there, no, not just an overall index of aberrations per cell. The need for detailed data is also more important because of non-uniformity in the classification of aberrations and disagreement on whether a small achromatic unstained gap in the chromosome are true chromosomal aberrations or not. To differentiate between the actual chromosomal damage and the one which is false or negative, we have to be able to differentiate. Gap should be quantified but not pulled with other aberrations. So same type of aberrations can be pulled, but the gap but they should not be pulled with other aberrations. So let's see what else we have in under this cytogenic chromosome aberrations only. In interpreting the results on the induction of chromosomal aberrations in cell culture, one must be alert to the possibility of the artifacts associated with the extreme assay conditions, because aberrations induced under such circumstances may not be of reflection of a chemical specific genotoxicity. We need to have a clear picture whether it is because of the test item what we are or is it because of the some physical artifacts or circumstances. Questionable positive results have been found at highly cytotoxic concentrations, high osmolality, and pH extremes. So this is very important. We are entering into highly cytotoxic concentration. The possibility that metabolic activation system may be genotoxic also warrants the scrutiny. Although excessively high concentrations may lead to false positive responses, the failure to test to sufficiently high dose levels may undermine the utility of the test. So whenever you are doing this assay, you have to choose the dose which are causing cytotoxic. See, therefore testing should be extended to a concentration at which there is some cytotoxicity. So you have to select the concentration which are causing some cytotoxicity, such as reduction in the replicative index of the mitotic index, the proportion of the cells in the division cycle. If a chemical is non-toxic, testing concentration should be extended up to a arbitrary concentration limit by a consensus of cytogenetics and genetic toxicology. The limit, what is the limit which is given, is 10 millimolar or 2 mg per ml, whichever is lower and has been recommended. However, this limit dose for the evaluation of human pharmaceuticals is how much? 1 millimolar. For chemicals, it is 10 millimolar 2 mg per ml, but for coming to pharmaceutical, they have given it as 1 millimolar. Okay, so you have to select cytotoxic concentration, but it should not be too much cytotoxic also, where it is giving wrong type of results. In vivo assay for chromosome aberration involves treating intact animal and later collecting the cells for cytogenic analysis. The main advantage of this in vivo assay is that they include mammalian metabolism, DNA repair, as well as pharmacodynamics. The target is typically tissue from which large number of dividing cells are easily prepared for analysis. Bone marrow from rat, mice, Chinese hamster is most commonly used. Peripheral lymphocytes are another suitable target. So first we have discussed about Chinese hamster ovary cells, then we are discussing about the intact in vivo test. Now we are discussing that is bone marrow from rat, mice and Chinese hamsters. Now we are discussing peripheral lymphocytes are another suitable target when stimulated to divide with a mitogen such as phytohemagglutinin. Effective testing requires dosage of the routes of administration that ensure adequate exposure of the target cells, proper intervals between the treatment as well as collecting cells, and sufficient number of animals are cells analyzed. So in continuation with this one, we'll also be reading about fluorescent in situ hybridization. An important development in this cytogenetic analysis is fish assay. So in which a nucleic acid probe is hybridized to complementary sequence in chromosomal DNA. Okay, you are hybridizing this nucleic acid probe. The probe is labeled with a fluorescent dye so that chromosomal location to which it binds is visible by this fluorescent microscopy, and the signal can be increased in strength by computer enhanced process. Okay, the use of whole chromosome probes probed during metaphase or interphase nuclei is commonly called chromosome painting. So in next picture we'll see what is that chromosome painting. The level of the sophistication has increased so much that all 24 different human chromosomes, 22 autosomes X and Y can be individually detected and can all mouse chromosomes. So let's look into this picture. This is the picture what we are discussing. Although fish is not routinely used in genotoxicity testing, it is valuable research tool for studying clastogens. Okay, fish assay, and is having a substantial impact on monitoring human populations for chromosomal damage. So if you look into figure 9.7, this one shows a chromosome painting and reciprocal translocations in human cancer cells. The regions of hybridization can be determined by use of these fluorescent antibodies that detect modified DNA bases incorporated during amplification or by incorporating fluorescent bases themselves during amplification. Another significant advantage of this fish method is that probes can be used with interface cells or chromosome, making any tissue potentially available for the analysis. Most recently, two types are there. M fish, that is multicolor fish, M band, that is multicolor banding, have been developed for defining these chromosomal abnormalities. Alterations in the tumor can also be detected to whole genome basis. The following example highlights the ability to construct breakpoint profiles of specific tumor types. One of the best known examples of this chromosome breakpoint is Philadelphia chromosome. So you might have come across this word, where translocation abnormality of this chromosome 22 containing a part of the chromosome 9 that is often found in chronic myelogenous leukemia and sometimes in acute lymphocytic leukemia. You can see in translocations the involvement of specific chromosome band that contains this oncogene or tumor suppressor gene is also known. Okay, so specifically if you see chromosome 4 painted with a probe while other chromosomes were counterstained in different color, you can see now one would see only two homologous of chromosome 4 in the color of the probe in the normal cell. However, if there are a translocation or dicentric chromosome and fragment involving chromosome 4, one would see these areas of fluorescence, one normal chromosome 4 and two pieces involved with chromosome rearrangement. Aberrations are detected only in the painted portion of the genome, but this advantage can be offset by painting a few chromosomes simultaneously with the probe of different colors. So from this hope you can clearly see this human breast cancer cell aneuploidy for some chromosomes and with reciprocal translocations. Okay, you can see the color switching along the chromosome, right? So by this techniques, no, we will be able to identify the chromosomal damage. So hope from this video you have learned about particularly the focus was more towards whatever the in the Clayson textbook, no, whatever they have given about the chromosome aberration under that heading in the genotoxicology chapter, no, I try to involve so that any question that comes around this particular concept we will be able to answer in the exam. Thank you so much. Be curious. If for any questions you can always write a email to arya.priyanka18@gmail.com. Like, subscribe and follow my YouTube channel Toxicology on Board. Thank you so much. All the best for your exam.